Hello, welcome to the 6th week of the class, starting September 26th. What we're going to go over is reiterate some topics discussed in Chapter 6 last week, as well as go over Chapter 7. Just a heads up of what's happening in the next few weeks. Next week, starting October 3rd, we're going to go over Chapters 8 and 9. And then that following week of October 10th will be your midterm. In the syllabus it said that we'd be having the midterm over chapters 1 and 10, but we're actually probably just going to get through chapter 9 material. So just, double check whatever slides I post for the weeks prior to that, because that's what I'll mostly be testing on. I'm going to post the test on that Tuesday and expect it turned in that Thursday. Just to remind you, all the cells of a multicellular eukaryotic organism can be divided into two types. There's the somatic cells, which are the cells forming the body of the organism. So my skin cells, my liver cells, my hair cells, like everything except for reproductive cells. And the reproductive cells are the sex cells, that is the gametes, which are sperm and egg. Somatic cells and reproductive cells use different methods of producing new cells. Alright, so the somatic cells use that mitosis method, and those reproductive cells use the meiosis method. The homework this week will be partly over meiosis. The homework last week was over mitosis and meiosis, so you need to definitely understand these two processes and how they differ from each other. Something common to both of them is interphase. So 90% of any cell cycle is spent in interphase when the cell is getting ready for cell division. And it's during that time that the cell is growing, that the organelles are doubling, and importantly that the DNA is replicating. So just before cells divide, the DNA molecule unwinds, and each half of it serves as a template on which the missing half is reconstructed. So the point is that at the end of the process of reconstructing the missing halves, there are two DNA molecules, each identical to the original DNA molecule, one for each of the two new cells after cell division. What those little A's are on that picture is those would be genes, so it would be a unit of heredity. The reason why you have a blue and a red and a capital A and a little a is you get one chromosome. Let's say that's chromosome 3, for example. So you get one copy of chromosome 3 from your father, let's say that's blue, and then one copy of chromosome 3 from your mother, let's say that's the purple one. Well, they each on the exact same spot on the chromosome carry a gene. That should be the same gene, but it may have some differences, right? So let's say it's the gene for hair color. Well, maybe that big A represents dark brown hair, but that little a represents red hair. So it's a combination of those two different alleles, is what we call them. Those two different alleles that, when combined, will equal the phenotype or the physical characteristic of those genes. Okay, so after interphase, you're going to have the DNA replication. You're going to see that they're attached at that point before they go through cell division. Remember that mitosis produces diploid cells, di-2-ploid sets. So we as human beings have 46 total chromosomes, but that's because we have two pairs of 23. 23 from our mom and 23 from our dad, right? So the di means that there's two sets of those 23. Whereas meiosis occurs in the reproductive cells, and the reproductive cells just have one set of 23. They're haploid, have half the number. Another thing you need to remember is that all of the cells for mitosis are genetically identical from the parent cell. So that one parent cell that's split into two daughter cells, those are now like exact clones of each other, basically. Sexually reproducing organisms have evolved a way to avoid the inevitable chromosome overload. Okay, so if every generation we kept doubling the number of chromosomes by putting a full set together from an egg and a sperm, we'd end up with thousands and thousands of chromosomes before not too many generations have passed. So to maintain a stable genome size, we use meiosis to have haploid reproductive gamete cells. Okay, so gamete cells have only one set of 23 chromosomes rather than two. And that in part is because meiosis, shown here on the right, has 2 rounds of cell division. And something I want to point out here that I think confused my other class is that your parent cell, in both the case of mitosis and meiosis, is diploid. So all of your sperm and your egg arise from a cell that is diploid. They are haploid after they are made, but the original cell is going to be diploid. And that's going to be covered some in your homework this week. This is just reiterating that parent cell is diploid, but then you have 2 rounds of cell division, so you have daughter cells that are haploid. Also, you have 4 instead of 2 in the case of mitosis. And all of them are genetically different after meiosis, whereas remember, mitosis, they were identical. Alright, so the important points of meiosis is that in addition to the haploid nature, is that there's genetic variation introduced. So at fertilization, 2 haploid cells, each with one set of 23 chromosomes, merge and create a new individual with the proper diploid human genome of 46 chromosomes. When the time comes to reproduce, this new individual, through meiosis, also will produce haploid gametes that have only one set of 23 chromosomes. With sexual reproduction then, diploid organisms produce haploid gametes that fuse at fertilization to restore the diploid state. It is in this way that meiosis maintains a stable genome size in a species. So you should be able to fill in this chart. You should be able to make a chart comparing mitosis and meiosis and know how many chromosomes in the case of a human are going to be in each, right? So mitosis, the type of cells were somatic cells. They are diploid, which means they have 2 times 23 chromosomes, so 46 total. Haploid is meiosis, so you have 23 total because there's only one set, and these are reproductive cells. This is a good YouTube video to watch to compare the two. I know that meiosis gets a little confusing for people because there's so many similarities to mitosis that they don't really understand, like, why do we end up with 4 haploid cells instead of 2 diploid cells? So this video will help you out, and then it's also part of your homework, so you'll watch it for that anyway. In addition to meiosis producing haploid cells, there's confusion with how genetic variation arises in meiosis. And that has to do with the way that chromosomes line up before they're split into 2 new daughter cells. Okay, so I didn't really spend a lot of time making you memorize all of the steps of mitosis. So remember, like, prophase, metaphase, anaphase, telophase, cytokinesis. I don't really care about you knowing all that minutia, but I do need you to know that those chromosomes line up. So interphase, the chromosomes duplicate, and then they line up in the middle of the cell, and then they're pulled to 2 separate ends, and then those 2 separate ends become 2 separate daughter cells, right? And the reason you need to know that is because it's the way that they arrange before they're pulled apart that leads to genetic variation in the case of meiosis. So you can see that here. Remember, there's 2 rounds of cell division. So in the top left, you had all of the blue line up on the left, but all of the right line up on the right. Okay, but then if you look at possibility 2, see how they lined up differently. Well, follow those chromosomes through those 2 rounds of cell division and see how those daughter cells end up with different combinations, right? So that can end up in genetic diversity. So if combination 1 becomes fertilized and becomes a baby boy, but then combination 4 becomes a baby boy, they're going to look different from each other even if they have the same mother and father. Alright, so now we're going to go into chapter 7. I'm going to focus on sections 1 through 5 and then 10 through 13. So not very many sections. Okay, so family resemblance is because your mother and your father pass on genes. I think everybody seems to know that, but you don't quite understand the mechanism of how that works or why that works. But it becomes pretty important. I mean, this is the basis for what makes you you, and it can be the basis for what diseases you may be at risk at, and your own children may be at risk at you passing on and them inheriting from you. So can a gene be cruel? Of course not, but if one could, consider this candidate. In humans, there is a gene for an enzyme called FMO3, which breaks down a chemical in the body that smells like rotting fish. Some unfortunate individuals inherit a defective FMO3 gene and cannot break the noxious chemical down. Instead, their urine, sweat, and breath excrete it, causing them to smell like rotting fish. Worst of all, because the odor comes from within their bodies, they cannot wash it away no matter how hard they tried. Called fish odor syndrome, this disorder often causes its carriers to suffer ridicule, social isolation, and depression. For those born with this malady, beyond their own suffering, there looms a scary question: will I pass this condition on to my children? They may also wonder how they came to have the disorder, particularly if neither of their parents suffered from it, right? So how do people pass on a disease potentially to their children if they themselves don't have it? Or how can your children have a disease that your grandparents had? Like, how much are your children at risk of getting it or not? How do you know? Well, we can know. For a lot of cases. Sickle cell is a really good example that I usually get a lot of questions about. So I'm going to talk more about that than about this FMO3 fish thing, because I've never met somebody with that, but I do know sickle cell is relevant, particularly in the African American community. Okay, so this is showing you some background for the inheritance of genes before I go into the example. So we just talked about this. You have your 2 copies of every single chromosome. So a mom has 2 copies of a chromosome and a dad has 2 copies of a chromosome in all of their body cells, right? The exception to that is the egg and sperm cell is going to have 1. Now which one it gets, that's a 50-50 shot. So a mom's chromosome and a dad's chromosome will come together after fertilization, and that baby will then have the correct 2 copies of that chromosome. But more importantly, we're not just talking about chromosomes, we're talking about genes. So genes are tiny units within those chromosomes that you need 2 copies of. Okay, so you get 1 copy of a gene from your mom, 1 copy of a gene from your dad. We call that different type of gene copy an allele. And it's these alleles that can encode for all the proteins in our body. So they can encode for a totally normal protein that looks correct and can do its job correct, or it can encode for a mutant protein that is all messed up and maybe can do its job but not as well, or maybe can't do its job at all, or maybe can't even be formed because it's so screwed up. Alright, so let's take the sickle cell anemia example. So everything starts with your DNA code. Alright, so you remember those nucleotides, those G's, those C's, those A's, and their T's, and it's showing you here that G's and C's match up and A's and T's match up. And then DNA gets transcribed into RNA, and then it gets translated into protein. I hope you remember that. So here on the left you see a normal DNA. So this is a codon, okay? Remember a codon is the 3 nucleotides that A codon encodes for A amino acid. So that GAG gets transcribed into RNA, and then that codon is read at the ribosomes during translation and says, hey, we need to have this glutamine amino acid here in the protein. Well, in sickle cell, a mutation in a single nucleotide changes what amino acid is going to be built into the protein hemoglobin. Alright, so instead of getting the glutamine that it needs, the sickle cell mutation is going to cause a valine to be put into the hemoglobin. Well, a protein's made up of lots and lots of amino acids, so it seems kind of crazy, but one tiny amino acid difference can make a huge change in the structure of that protein. So in the case of hemoglobin, which is what fills up a red blood cell, it changes the entire shape of the red blood cell because the protein's job is to look a certain way to give a certain structure to those red blood cells. Red blood cells have evolved to look a specific way so that they can get through the blood vessels properly and then carry oxygen to all the body parts that they need to in an efficient manner. Sickle cell is a sickling shape of your red blood cells. The hemoglobin has that mutation, it doesn't form right. It changes the shape of the red blood cells from those flat discs to these sickled cells that end up getting stuck in the red blood vessels and not flowing to all the parts of your body that it needs to be flowing to. And it can be deadly. Okay, so what if a dad is a carrier for sickle cell and a mom is a carrier for sickle cell? What do I mean by carrier? I mean that they have a mutant allele. Because remember, they have two alleles, right? So one of their alleles is mutated, but they don't have the disease themselves because one of their alleles is normal. All right? So dad has one normal allele for hemoglobin and one mutant allele for hemoglobin. Mom, her two different gene alleles would be the same. One that's normal for hemoglobin and one for mutant for hemoglobin. So what are the chances that their kids will have the disease? You have to think about how mom and dad are only going to pass on one allele to the children apiece. And then that combination of one from mom and one from dad gives those kids those two, right? So it depends on which allele they pass on. And that's what this chart is showing you here. So depending how those alleles get passed on, mom's going to have eggs that have either the normal allele or eggs that have the mutant allele. If an egg with the normal allele, represented by a big R here, mixes with a sperm from dad with the normal allele, then you have an unaffected child. So there's a 1 in 4 chance of that. If a normal egg from mom mixes with a mutated sperm from dad, or the opposite, if a mutated egg from mom mixes with a normal sperm from dad, you'll have carrier children. So those children will have one normal and one mutated allele. So there's a 2 in 4 or 50% chance of that. But in the unfortunate chance that a mother's mutated egg is fertilized by a father's mutated sperm alleles, then you get both those mutated alleles together and then that child will have sickle cell disease. And that's a 1 in 4 chance or 25%. Later on we'll do Punnett squares to show you how I got those chances worked out. It's pretty easy to figure out. So then the question people always ask me is like, if mom and dad both have a mutant allele, why aren't they sick themselves? Why do you have to pass on 2 mutated alleles to have the disease? Like, why isn't one enough? And we will talk about that later. Alright, let's change it up a little bit. What if dad has the disease, which means he has 2 mutant alleles and no normal hemoglobin alleles, and mom is still a carrier? Then what are the chances that kids will have the disease? I think intuitively you think you will know the chances are greater, right? Well, let's work out why. Okay, so instead of these R's representing alleles, you're seeing the actual image for a sickled cell here. So these are all the possible combinations, all right? So there's the possibility of a sickled cell from dad every single time. So it's going to be the determining fact, because he has a disease, right? Both his alleles are mutated. What's going to be the determining factor is if mother passes on a normal allele or a sickled cell allele. So no matter what, their offspring is going to get at least one sickled cell allele because dad is 100%. So they're at least going to be carriers for sickle cell. But then the other 50% is they got that mutated egg from mom, so then they have a 50% chance of having sickle cell disease. So how could 2 normal parents have a child with sickle cell disorder? I forgot to change FMO3 to sickled cell, so just read that as saying sickle cell. So go ahead, pause this, take a moment, and read all those answers and pick one. The answer is number 4. So that's the example we worked through first. You're a normal parent because you're a carrier, which means you have a mutated allele you can pass on, but you don't necessarily have the disease yourself because you have a healthy allele. So if that doesn't make sense, go back and review that. Offspring resemble their parents because they inherit genes from them, and genes are the set instructions for making proteins. So Genotype, your genes, encode your phenotype, your physical characteristics or traits. Those are two terms you should know. Some traits are controlled by a single gene. The two examples we just talked about are, a lot of times they're not. We'll talk about that in the later sections of this chapter. But here's another term you should know, heredity. So usually it's more complicated. It's biology, so it tends to be more complicated. There are multiple gene traits. It's not usually just one gene equals one phenotype, one physical trait. And we'll get into that later. But for the cases where there is a single gene that equals a single trait or phenotype, that's a straightforward pattern of inheritance. All right, that's the most straightforward. And we learned about this pattern of inheritance from a guy named Gregor Mendel, who was an Austrian monk. So what do parents give their offspring that confers similarity? Like, what is actually passed on? We know it has something to do with the sperm and the egg, and now you know it has something to do with DNA, but they didn't used to know that. That's a pretty recent discovery. If you remember when I talked about them discovering that DNA was the unit of heredity. Like, what time frame was that? Do you remember? You should probably look that up. So how is it that a physical entity is inherited, and how is it passed from parent to offspring? It wasn't until the mid-1800s that any real headway had been made on these questions. At that time, Gregor Mendel, a monk living in what is now Czech Republic, began some studies that not only shed light on these questions, but practically answered them completely. So before they even knew about DNA, Gregor Mendel almost completely worked all of this out with his experiments. Mendel understood the essence of the genetics puzzle and set out to piece it all together. When Mendel turned to these questions, there were no obvious answers. At the time, there was an idea that had been popular since the late 1600s that suggested that an entire pre-made human, albeit a tiny one, was contained in every sperm. So this is called homunculus hypothesis. So before Mendel knew anything, before anyone knew anything in the 1900s about DNA, there was a few, crazy hypotheses running around about how do babies come about. And one of them was that the guy just, already created a human being and the women were just incubators in a way. Right? So they didn't really understand that women and men contributed equally genetic material because they didn't know about genetic material. So just to reiterate, this is not how it works. I'm trying to show you some of the explanations that are obviously very wrong that people tried to use to explain things back during Mendel's time. It was kind of amazing that he came to the conclusions he did given the context he was in. Mendel's success stemmed from how he applied the tried and true process of methodical experimentation and scientific thinking. If you've heard anything about Mendel, you probably remember pea plants. The pea plant was the organism that he studied. So monks, he's a Christian monk, they tend to be self-sufficient. They live in isolation. They tend to make their own food. They tend to do their own gardening. They make beer and wine that they may sell as a source of revenue. So he was a monk, theologian, but also a gardener. And he spent a lot of time with plants. He spent a lot of time with pea plants. And he noticed that he could cross them, right? So if you're a gardener and you want to make a certain color plant, you cross two different colors of plants, and then you see what happens until you finally get the color that you want. So he was messing with stuff like that. And he looked at things like the flower color, the pea color, like peas have a color to them. They're not just always green. And then the shape of the peas themselves. So he made observations, and then he came up with a hypothesis, and then he did some experimentation to test it. Okay? Shown here. The egg and the sperm only have one copy apiece. And those are gametes. At fertilization, offspring receive from each parent one allele for each gene. okay? So these are heterozygous pea plants. So these are the parent plants. This would be like the mommy and daddy plant. And they're heterozygous because they each carry 2 different alleles. Alright, we call that heterozygous. It's just the word you gotta know. One is for white flower color and one is for purple flower color. And they each contain one of each. Now, why do they appear purple? Why is the mom and dad plant purple if they contain a white and a purple gene? Well, blending is not usually how genes work. Like in the case of sickle cell, one dominates over the other. So a dominant trait masks the effect of a recessive trait. Okay? So if you have a healthy hemoglobin protein, that's going to dominate over the mutated hemoglobin protein. So that's why people that are carriers don't have sickle cell disease, because they have a dominant wild type allele. So here the dominant allele is purple. So these plants appear purple even though they carry a white allele as well. So meiosis happens and each gamete gets 1 copy of each gene. Alright? So that means it's got a 50-50 shot of getting a white allele from the mom or a purple allele from the mom. Also, it has a 50-50 shot of getting a white allele from dad and the purple allele from the dad. So if you follow all of those crosses, you can see all the combinations that happen, just like we did with the sickle cell. Going from left to right, so you have a 1 in 4 chance of the plant having 2 white alleles. So those are both the recessive alleles, so it's white. The 2 center ones have 1 purple and 1 white allele, so those are carriers. Those are heterozygous. So they appear purple because of that dominant trait that they have. And then on the right, it's very straightforward. You have 2 dominant alleles, so you're just going to be that color. Know these terms on the bottom. So heterozygous I already went over. Homozygous means, homo, it's a prefix, that means same. So homozygous means they have 2 of the same alleles. Those 2 alleles could either be dominant or recessive. So if you have 2 dominant alleles, you have homozygous dominant genotype. If you have 2 recessive alleles, you have homozygous recessive genotype. And then if you have 2 different types of alleles, you're heterozygous genotype. So rather than passing on a trait itself, each parent puts into every sperm or egg a single set of instructions for building the trait. That is the instruction set that today we call a gene. Offspring receive 2 copies of the instructions for any trait. Often, both sets of instructions are identical and the offspring produces the traits according to those instructions. Occasionally, though, each parent contributes a slightly different set of instructions for the trait. These alternate forms of a gene are called alleles. Know that term. So, for example, in pea plants, there are 2 alleles for flower color, a purple flower allele and a white flower allele. Each is a different allele, but because both alleles still specify instructions for producing flowers, they are both genes for flower color. Some gene, flower color gene, different alleles, purple and white. The trait observed in an individual depends on the 2 copies of the gene that it inherits from its parents. When an individual inherits 2 copies of the same allele, it is homozygous and shows the traits specified by the instructions embodied in those alleles. When an individual inherits a different allele from each parent, it is heterozygous. Dominant and recessive alleles are defined by their action when they are in the heterozygous state. If one of the alleles is fully apparent in the individual while the other is not, the first is called the dominant allele and is said to mask the effect of the other allele, which is called the recessive allele. I want to point out here too, I find this very strange that people think this, but I get asked this by my students all the time. Dominant and recessive is not a gendered thing. Like, you're not getting all your dominant alleles from your dad and all your recessive alleles from your mom. Your mom can pass on a dominant allele just as much as your father can. Dominant just means it's the trait that's going to mask any effect that a recessive allele would have. It's not gendered at all. Alright, so these are the 3 different genotypes again. We tend to express the dominant allele with a capital letter and then the recessive allele with a little letter. Okay, so that homozygous dominant, big R, If we look back at, if we take the example of purple versus white, that would be a purple plant. If we take the example of sickle cell, that would be a normal individual with normal hemoglobin, normal red blood cells. If we look at the heterozygous, same phenotype. It would have purple plant because there's that dominant allele, or it would have no sickle cell because of that dominant allele. Homozygous recessive, that case you will have a white plant because there's no purple allele to dominate it, so it gets to just be white. Or in the case of sickle cell, this person would have sickle cell disease because the hemoglobin would all be mutated. So remember, originally hemoglobin has a specific set of instructions to make it correctly. So what happened to make a messed up version is you had a mutation in that DNA nucleotide. Like the wrong nucleotide got put in at some point, which resulted in the wrong amino acid. So these things these mutated alleles can persist from generation to generation only because the dominant allele takes over and masks any ill effects that it may have. And that's how genetic variation comes about, and these different types of alleles come about, are these mutations. Only if a person has 2 mutant versions of the hemoglobin gene, then that person's genotype is, pause if you need to, answer is number, hold on one second, homozygous recessive. Which choice below represents a cross between a heterozygous plant and a homozygous plant? So here you'd have to draw them all out and you'd have to figure out which percentages go with which, and I'm going to show you how to do that using Punnett squares in your homework. The answer would be 3 though. In order to get both purple and white offspring, the purple parent would have to be heterozygous. White is homozygous. Alright, these are take-home messages. So observing an individual's phenotype is not sufficient for determining its genotype. Why is that? Well, think about that carrier thing again, or the correct term is a heterozygous person. Right? So if you look at somebody who doesn't have sickle cell, do you know if they're a carrier or not? No, you don't. You don't know their genotype by looking at that normal phenotype. If you look at a purple plant and you say, oh, that's purple, do you know its genotype based on that purple phenotype? No, you do not, because it could either be homozygous dominant, big R, or it could be heterozygous, big R, little r. You have no idea. Things are not always as they appear. The skin coloration, take skin coloration for example. Humans and many other mammals have a gene that contains the information producing melanin. If you've never heard of melanin before, it's one of the chemicals responsible for giving our skin its color. Unfortunately, there's also a defective non-functioning version of the melanin gene that is passed along through some families. An individual who inherits 2 copies of the defective version of the gene cannot produce pigment and has a condition known as albinism, a disorder characterized by little or no pigment in the eyes, hair, and skin. It is impossible to tell whether a normally pigmented individual carries one of the defective alleles just by looking. One would need to do a genetic analysis to discover this information. This is just reiterating the phenotype is the outward appearance and the genotype is the underlying genetic information. So you can't tell if a deer with normal skin pigmentation is homozygous dominant or heterozygous. All you know about its genotype is that it isn't homozygous recessive, because then it would be an albino. So how do we analyze and predict what someone's genotype is? Well, in the case of plants, you can do crosses, look at the offspring, and then you can go back and use deductive reasoning to see what the parents were. So we assign symbols to represent the different variants of a gene. I've already talked about this. So we use an uppercase letter for the dominant allele and a lowercase letter for the recessive allele. So to analyze and predict the outcomes of crosses, first we assign those symbols. We represent the albino individual as r because it must carry 2 copies of the recessive allele. An individual that is pigmented must be big R or big R. If we don't know which one of the 2 possible genotypes a pigmented individual is, we can write big R and then a blank, where blank is a placeholder for the unknown second allele, but we're not going to really talk about that. And I just keep using R to be consistent. It could be any letter. Like, you'll see it in test questions or in the book as any letter. Usually they pick a letter that they think best represents the trait they're looking at. So maybe someone else would think, oh, I should use a big P and a little p because I'm talking about purple flowers. Or someone will use a big H and a little h because they're talking about hemoglobin. All you need to know is the big letter is the dominant one and the little letter is the recessive one, and you need to know those 3 different types of genotypes: the homozygous dominant, the homozygous recessive, and the heterozygous. So here is an actual cross. So you'll do this in your homework, it'll make more sense. But here's one example for you. On the left, we have the mom, and on the top, we have the dad. We have their genotype, okay? So the dad has 2 alleles of this. We're going to use the melanin example. So there's 2 alleles for melanin. One is big R, which means normal melanin. One is little r, which means a mutated melanin. On the left down, mom is the same. She carries that little r, but she also has a big R. So they're both heterozygous, okay? So for the cross, what we do is we put 1 letter from the mom and 1 letter from the dad in each square. Because remember, you get an egg and a sperm, each carrying 1 allele, matching up to give the offspring 2 alleles. Okay, so match up where all those got drawn in. So you can figure out what percentage of the offspring will have what genotypes, and then you can figure out what percentage of the offspring will have the normal skin pigmentation versus albino phenotypes. Okay? So you have 1 in 4, or 25% of the offspring had big R. What is that called? Think about it. Homozygous dominant. You have 2 out of 4, or 50%, is the big R, little r genotype. What is that called? Heterozygous. And then the leftover 1 in 4, or 25%, is little r, which is homozygous recessive. We can then figure out those phenotypes, right? Because anytime there's a dominant allele around, you're going to have the dominant phenotype, which is normal skin pigmentation. So 3 out of 4, or 75%, are normal skin pigmentation, and 1 in 4 are little r, which means that they have the albino phenotype. So go ahead and try and draw your own Punnett square right now. Pause the video. It might take you like 5 minutes to figure it out, but try and figure it out. What is the likelihood that a white offspring will be born to the following parents? Okay, so what they're using here to represent melanin is an M. I just used an R. It's the same principle. So you don't need to know which one's mom and which one's dad. Just take those 2 different genotypes and line one up on the side, left-hand side, and one up on the top. So on the top of one, you should have a little m, and then a little m, and then on the left side, you should have a big M, and then a little m right under it, and then go ahead and match up in those 4 squares. Think of these are called ratios. Just think of them as fractions. So instead of those colons, just put a slash, right? So that's saying the likelihood of a melanin offspring is 4 in 4, or 3 in 4, or 2 in 4, or 1 in 4, or 0. It's just a different way to represent a fraction. The answer is number 3, 2 in 4. This take-home message. So for a particular trait, an individual may carry a recessive allele whose phenotypic effect is masked by the presence of the dominant one. Alright, and we use Punnett squares. Okay, all of those examples we're talking about 1 gene equals 1 phenotype. But that's not always the case. I don't want you to think that's always the case. Like cancer. People always ask me about cancer. Like, my grandpa had cancer. Am I going to have cancer? You know, it's more complicated than your grandpa had this one mutation and you're definitely going to have it. Because it's multiple genes that affect cancer and environmental effects on top of that. So it's very complicated. Whereas something like sickle cell, we have one single mutation. We can track it down. One gene equals defective melanin or not. Or, hemoglobin or not. Blood type is an interesting situation. So I've been talking over and over about how every gene has 2 alleles, right? Well, in the case of blood type, it's more complicated than that. So what is your blood type? It can be either O, A, B, or AB. Each of these different blood types indicates something about the physical characteristics of your red blood cells and has implications for blood transfusions, both giving and receiving blood. I'm not going to talk about if you're A negative or positive, what those positives and negatives mean. I'm just going to talk about the alleles itself. But I don't want you to get confused and think that you have 3 alleles running around for blood. You don't. You can only ever have 2 alleles for every gene because you only ever get one from your mom and one for your dad of a chromosome. And those alleles are on chromosomes, okay? But what multiple allelism is, like in the case of blood type, is when a single gene has more than 2 allele types. Okay, so you're going to get 2, but there's more than 2 out there in the gene pool. Blood type, for example, has 3. Inheritance of the ABO blood group is the simplest example of multiple allelism because there are only 3 alleles. You can call these A, B, and O. The A and B alleles are both completely dominant to O. Alright, so we're back to that dominant recessive thing. So O is recessive, A is dominant, however, B is also dominant. So we call A and B codominant. So in the case of having A and B together, so if I get an A allele from my mom and a B allele from my dad, those are going to be codominant. I'm going to have both expressed on my blood cells, which is very unique. But if my blood type is A, what that means is I have an A from my mom, but I actually got an O from my dad. But it's presenting on my blood cells as A because it's dominant over that recessive O. Same thing for B. And then to be O, you have to have 2 recessive alleles. Okay? So this is showing that. Definitely pause here. Try and work your way through this and understand. But type on the bottom left, type A would be you either have 2 of those A alleles or even A, and little I represents an O. Type B, same thing. Type AB would be that codominance, and then type O would be of those 2 recessive alleles, little I, which are the O type. Alright, if you are type O, or if you've ever talked about like blood transfusions, you've probably brought up the like type O can be a universal donor or not. It gets kind of confusing, but this is the reason why. Individuals with only B antigens on their red blood cells produce antibodies that attack A antigens. But blood type O individuals who have neither A nor B antigens on their red blood cells produce both antibodies that attack both A and B. So individuals with blood type AB don't produce either type of antibody. From this information, we can deduce which blood types can be used in transfusions. Okay, so antigens are things present on the surface of the red blood cell, and they recognize antibodies that are floating around in your bloodstream. Antigens attack things that are different from them. That's just how they work. So if you have type A blood, you're going to attack type B blood. So you don't want to give somebody with type A blood a transfusion of type B. So they can only receive type A blood or type O blood. Type B would be like the inverse. Type A and B is going to recognize both A and B. So they can receive from A or B, or an AB, because those are both, that's the exact same thing, or an O. O doesn't have any antigens, antibodies for it to attack. That's why it's a universal donor. However, type O can't really receive blood from anyone except for type O, so type O is kind of in a crappy situation. So if you have type O blood, you can give it to anybody, but you can't necessarily receive it because you don't have antigens on the surface of your red blood cells. So anything that's from A, B, or A plus B is going to appear foreign to you, and your body won't accept it. You'll reject that blood. So if you're AB, you never have to give away your blood, but you can get blood from everybody. So you hit the blood jackpot. But if you're type O, you really got the short end of the stick, because you can donate your blood to anybody and be nice to everybody else, but you can't really get blood from most people. It's rare. Okay, so that's an example of multiple allelism. So know what multiple allelism is. We're not going over that because you're not in a class. An example, okay, widow's peak is a dominant phenotype. If you have a widow's peak, what is your genotype? Think about it. Write it down. Pause. Okay, the answer would be number 4. Because remember, the widow's peak is dominant. That means it's a big W. You can either be homozygous dominant genotype and have the widow's peak phenotype, or you could be heterozygous genotype and have the widow's peak phenotype. Because if that dominant allele is around, then you're good to go. So number 3 would be the only time where you don't, and that would be a straight hairline because you have 2 recessive alleles. Okay, another exception to that one gene equals one trait thing is multiple gene traits. And examples of that would be like height, skin color, things that you can see a spectrum of variety with. So it's a phenotype or a trait that is influenced by many different genes. So skin color is one of those. That's why we don't really see like you're all white or you're all black. Like, no, you're a variety of colors. You can be much, much darker. You can be much, much, much lighter. You could be somewhere in the middle. And that's because there's a variety of genes. And for each gene, there's 2 alleles, so there's tons of different possibilities, which is what those little domino-looking things are showing you on the top. But my point here with the graph is anytime you see a bell curve, which is that sort of a distribution on a graph, that means that it's a polygenic trait. Okay, so know the term polygenic. That means that there's many poly genes, genic, to one trait, which in this case is skin color. Height would be a similar situation. So you don't really have a classroom to look around, but which trait is likely multigenic? All of the above, right? Because those all have a wide variety of phenotypes in our society. Okay, well now kind of the flip side of that polygenic thing is pleiotropy. So sometimes one gene influences multiple traits. That would be pleiotropy. Whereas polygenic would be multiple genes equal one trait. Alright, so that one might be hard for you to remember. I would definitely write that down. I remember polygenic because of many genic genes. So I remember there are many genes to one trait. Here I just remember it's the opposite one. There's an example of pleiotropy in your homework that you'll go over, and there's some in your book as well, so you can read up on those. And then lastly, all the examples we've talked about are going to be genes for the chromosomes that are not your sex chromosomes, okay? So we have 46 chromosomes total, 2 sets of 23, so that means there's chromosomes 1 through 23, alright? There's a chromosome 1, a chromosome 2, chromosome 3, chromosome 4, blah, 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 all the way through chromosome 23. Okay, well your chromosome set number 23, those are your sex chromosomes. Those are the XY that determine if you're boy or girl. But numbers 1 through 22, those are for everything else. Okay? So that sex chromosomes usually don't encode for very many things that aren't directly related to reproduction and gender, but there are a couple of things on those genes, on those chromosomes, that are important for other traits. So color blindness, for example, is on there. Hemophilia, for example, is on there. So there are rare exceptions where you have sex-linked traits. Okay? So that what that means is that they differ in their pattern of expression in males and females. One example is the red-green color blindness. So if you're a female, you have 2 X chromosomes. One X is always from your mom, and then the other X you got from your dad. If you're a male, you have XY, sex chromosomes. The X you always get from your mom, and the Y you got from your dad. Okay, so women always have eggs with an X in them to give to their offspring, but sperm have a 50-50 shot of an X or a Y, so the sperm determines gender. Anyway, women have 2 X chromosomes, whereas men only have 1. So a lot of times with these sex-linked traits, they're going to pop up a lot more in men because they only have 1 X chromosome. So if they carry a recessive allele on the X chromosome, they don't have another copy to cancel it out. So in the X chromosome in humans, there is a gene that carries the instructions for producing light-sensitive protein in the eye. And these pigments make it possible to distinguish between the colors red and green. There is a rare allele for this gene that produces a not functioning version of that protein. Having some of the non-functioning protein is not a problem, as long as the person carries another normal version of the gene that is dominant, okay? But this poses a problem for men because they have only 1 X chromosome. In contrast, females inherit 2. So if a man is colorblind, did he inherit the condition from his mother, his father, or both parents? Well, really he inherited it from his mom, but he doesn't have a copy from his dad to cancel it out. Okay? So a sex-linked trait is carried on the X chromosome. It's pretty much always carried on the X chromosome, is what you need to know. Women carry 2 copies of X, men carry an X and a Y. That's what you also need to know. Okay? So we're using that big R, little r again to show dominant and recessive. So let's say big R means you are not colorblind because you make all the proteins you need to see all the colors. But little r would mean you have the recessive allele for that mutated protein, so you can't really see some of the colors. Okay, so for a male to be colorblind, they must inherit the colorblind allele from their mother. But for a female to be colorblind, she must have received one from both parents. So that's why you'll meet more colorblind men in your life than women. Like, it's pretty rare to meet a colorblind woman. I have only ever met one, but I have met at least a dozen colorblind men. So approximately 7 to 10% of men exhibit red-green color blindness, while less than 1% of women are red-green colorblind, and this is the reason why. Which parents below would have the possibility of producing a daughter who is colorblind? Pause it and think about it. Okay, the answer is number 4, and here's why. So if you drew a Punnett square, you absolutely could. You could just draw an X with a big R above it on the tops versus the sides. Okay, so like the dad would be XY, he can go on the top, and then the mom would be XX, she can go on the side. So the combinations here for number 1, you're either going to be XX female with 2 dominant R alleles, which means you have normal vision, or you're going to be an XY male with a dominant R allele from your mother and have normal vision. In the middle, a male is going to be X big R from his mom and then a Y from his dad, so he has normal vision. A female would be a carrier, though, because she gets that X from her dad with the recessive and X big R from her mom, so she's heterozygous. Number 3, the female could either be a carrier because she gets the X little r from her mom and the X big R from her dad, or she could be homozygous dominant and get the X big R from her dad and the X big R from her mom. In the case of the male, he has a 50-50 shot of being colorblind, okay? So he gets the Y from his mom and then the Y from his dad, and he gets either the X big R from his mom and he has normal vision, or he gets the X little r from his mom and he has color blindness. But the question asks about a daughter. So 4 is the correct answer. If a daughter gets the X from her dad, it's definitely going to have that recessive allele on it, right? But then she has a 50-50 shot of getting the 1 from her mom with that recessive allele on it too. So the female could either be heterozygous or she could be homozygous recessive, 50-50 shot, in which case she's colorblind. And I think this is the end of the slide, so next week we will pick up with Chapter 8.